Ladies and gentlemen, may I introduce Dr. Jeffrey Goldberg.
It's such a pleasure to be here, to see so many familiar faces, to meet new ones. I can't share enough about how meaningful it has been to me to watch Tom and the foundation board and the foundation staff, who are one after the next, always committed, doing the right thing, supporting our field, helping all of us push the field forward. It's been even twice in this past couple of years that I've called Tom up and said, hey, I know about this other faculty who's doing amazing work, and they just need like one more ounce of support. And Tom digs around and finds someone to do that. So just thank you to the Glaucoma Research Foundation. It is the model for what we want to do to bring vision protection and vision restoration forward. And I get to see Tom at events like this, but the main place I get to see Tom is multiple times a year when we're sitting next to each other in the audience at the glaucoma meetings, hearing about the science and your commitment to hearing about the science and always leaning over and whispering a really good question. It's awesome. It's awesome. So Tom, thank you. And thanks to all of the foundation staff. It's the best group to get to work with year after year. I don't want to belabor this evening too long, but I am inclined to share one short story before sharing some of the science that we're getting into. When I was a graduate student, Christopher Reeve had his horseback riding accident, and that led to a real influx of support for spinal cord injury research, which at the end of the day is really about regenerating the central nervous system. It's the exact same problem that we're trying to solve in the optic nerve, as we'll talk about in just a minute. The Christopher Reeve Foundation, at its pinnacle, was also an excellent supporter of the scientific research. And at one of their annual meetings, they had a kind of a town hall on the Saturday night in the middle of the meeting, where they invited the scientists at the meeting, but they also invited spinally injured patients, their family member caretakers, nurses who work in that area, to come together. And the main message that I, as a junior graduate student, took away from that was the message from our patients, who really said, hey, you don't have to make that mouse walk perfectly again. If you could just help us get one step better, if you could give us a little bladder control, a little hand strength back, we would be in your clinical trials. Don't wait. And it really made a deep impression on me that I feel like I've really carried forward now as an ophthalmologist and vision scientist, really carried forward, and I think really matches very much the ethos of this foundation, which is how do we make the advances, but also how do we bring them forward into patient care? Let's not forget those patients sitting there in front of us. So with that in mind, they asked me to talk about the future is now. How are we pushing forward the science? How are we bringing that science out of the laboratory into the clinic? And not just about the eye pressure lowering, which of course is important, but how are we going to protect vision? How are we going to restore vision? How are we going to bring back that vision for our patients? Glaucoma still is the #1 cause of irreversible vision loss in the world. And so that's after glasses, if we could just supply more glasses. That's after cataract surgery, if we could deliver more cataract surgery to impoverished areas around the world. After those two, which are solvable at scale, glaucoma is really our biggest problem in eye care. #1 cause. We call it the silent thief of vision, as this group knows, because it tends to hit your peripheral vision where you don't even notice, and your central vision lasts. And this is a large, growing global challenge. Of course, here we're pretty focused on the almost 6 million US patients who have either glaucoma or ocular hypertension, and the 170 million patients over age 40, people over age 40 who should be screened for glaucoma. But that doesn't even touch the 800 million people throughout Asia who should be screened for glaucoma. And so this is a large global challenge. When we do community surveys and check people just, walking into the supermarket, we'll figure out who has glaucoma, and it'll turn out that 50% of those people didn't know they had glaucoma, right? But 100% of people with glaucoma don't know if they're going to get worse. And we really need to solve for that too. So how are we going to solve some of these unmet needs of glaucoma? Now again, we're always looking for better ways to lower eye pressure, but we do have a lot of medicines, we do have a lot of surgeries, we've got lasers. Not to say we're done there yet, but I'm not going to focus there at all tonight. We need better biomarkers. We need better ways to improve the measures of risk, diagnosis, progression. We need to be able to say to a patient, not just, hey, you've gotten worse over the last 3 years, you're losing vision, let's advance your therapy. We need to be able to say to the patient sitting right there, that moment in front of us, your retina is unhealthy, your optic nerve is unhealthy, you're not getting adequate treatment. I'm going to advance your therapy today so you don't lose vision in the next 3 years. And then we need better treatments, and these treatments need to go beyond eye pressure. We need treatments like neuroprotection, which is how do we keep the cells in the retina and their fibers carrying all that vision down the optic nerve back to the brain? How do we keep them from degenerating further? That's neuroprotection. How do we induce or encourage regeneration? Just like we want to regenerate a spinal cord so a person can walk again, we want to regenerate the optic nerve so a person can see again. And then we've taken a term from the Alzheimer's literature called neuroenhancement. And there the idea is to take your sick but not yet dead retinal cells and give them a booster shot, get them firing on all cylinders again so that they can survive but also function better and actually improve your vision. So we've got to take a couple of really important steps, and I'm going to give you just a little taste of each of those. First of all, we cannot ever underestimate the importance of discovery science. We've got to understand the pathophysiology of retinal ganglion cell degeneration. If we don't understand why there's no regeneration, we can't figure out how to induce regeneration. If we can't understand why the retina and optic nerve are degenerating in glaucoma, we can't figure out what to replace or give back or help to get that to stop. And here we have a very clear window to help our patients, because it turns out that in diseases like, for example, glaucoma, where the eye pressure might be raised early in the disease, there's all sorts of pathophysiologies that are happening that are propagating the disease. And we've learned a lot about those over these last years. In fact, the Catalyst for a Cure Group 1 did some really formative work understanding why glaucoma kills the optic nerve. At some point, the axon in the optic nerve gets actually damaged, physically damaged. But it's only some point later that the retinal ganglion cells actually die. That happens later in the disease. So there's window between dysfunction and death. And that window is our opportunity to intervene to save vision in glaucoma. Now, when we, the clinicians, look in the back of your eye, this is the optic nerve, and this is an optic nerve that's quite damaged by glaucoma. That thin rim of orange around the center is where all the axons are supposed to leave the retina and go back down the optic nerve back to the brain. This particular patient has lost most of those axons, most of that tissue. And so, again, we really need to focus our understanding, our targeting of the retinal ganglion cells and the optic nerve. So the retinal ganglion cells, that's kind of where the action is, part one. These are the cells in the retina that collect all the light information. They send that axon fiber down the optic nerve, carry all the vision back to the brain. They're the ones that are dying in glaucoma. And we've learned a lot about why they're dying. What are the molecular pathways? What are the genes and molecules involved? And that's leading us into opportunities to treat those retinal ganglion cells so they don't die. And here it turns out there's a lot of shared biology between what's going on in our eyes in glaucoma and our brains if we have other neurodegenerative diseases. This retinal ganglion cell is actually stained to light up the mitochondria. Those are like the power energy source organelles inside cells. And they're filled with there, but it turns out that when you disrupt these energy source mitochondria, you get neuronal degeneration. And that happens, as pictured on the left here, in Alzheimer's disease and Parkinson's disease and other forms of optic atrophy. It happens throughout the brain. And what happens is those normal-looking, here colored green mitochondria, they start to break up, they fragment, and then the cell is not getting all the work done that it needs to get done, and that leads eventually to its death. And these axon mitochondria are highly regulated. Here we've taken pictures of the adult retina on the bottom, but also the developing retina. And through development, these mitochondria are highly regulated. And it turns out that these mitochondria is where a lot of that metabolism is happening inside the axon and where a lot of the action is. And I'm going to come back to that because we're figuring out ways to treat that as we move into clinical trials. The other thing is that neurons in the central nervous system, including in our eye, they need to be next to their neighbors and they need to get good growth signals. And these growth signals are called neurotrophic factors, and they're in our bodies, including in our retina, and they have a kind of an alphabet soup of acronyms like nerve growth factor, NGF, and ciliary neurotrophic factor, CNTF. And it turns out, we discovered, that you can't just give more growth factors. You can, it helps. But it turns out that if you depolarize and give electrical activity to neurons, the two come together and it becomes like a 1 + 1 = 4 effect. They synergize to help each other. And so we're testing these now in animal models where we've made the discovery, but as you'll see in a minute, moving these into human testing as well. So we've learned a lot about the molecular pathways that are keeping the neurons alive, but it turns out that there's another really key population of cells. I mentioned here's the optic nerve, and it turns out that's kind of actually where a lot of the action is in glaucoma. We know that glaucoma spreads in a way that anatomically conforms to the anatomy of the optic nerve, and there's a lot going in the optic nerve. Actually, if you take out all the cells of the optic nerve, there's these collagen beams. It's kind of like the structure inside a building holding the walls together. And that's what that looks like when you zoom in on it. And those beams are covered in support cells called astrocytes. If you kind of dissect the optic nerve, that's kind of where that action is happening as it dives down in. And those cells, it's like where all the transport is being regulated. When the eye pressure goes up, as happens on the right side of these pictures, when the eye pressure goes up, you actually block, right at the optic nerve head, you block the transport, the signaling between the eye and the brain. You shut it down when the eye pressure goes up. And so again, we know that the optic nerve head is where a lot of that action is. And we know that there's this cell type there called astrocytes. These are support cells. They're called astroglia. Glia actually means glue, and it was thought for a long time that they just kind of help glue the nervous system together. But it turns out they are really part of the action, and this has been relatively new discovery space. For a long time, we understood that astrocytes in glaucoma and other degenerative diseases can be detrimental. They react in such a way that they start spitting out negative factors that they sort of actively kill the retinal ganglion cells. But it turns out we've only really just begun to understand in some of this much more recent work that there's another population of reactive astrocytes. These are the good, beneficial reactive astrocytes. And for a long time, we didn't even have a way to identify which was which. Everyone actually thought they were all bad. It turns out there's some good, beneficial reactive astrocytes, and we've been studying these recently, and it turns out that this might be another whole way to develop therapeutics for glaucoma, for our human patients for glaucoma. Now, what's it going to take? We got to figure out how to target those cells, and we got to figure out what to deliver. Well, we've struck upon some really great findings solving both of those problems. First of all, we've discovered a new way to do gene therapy where the gene therapy goes just to the optic nerve head astrocytes. They're labeled in green in this picture. If we flatten out the retina, here's the retina kind of splayed out on our petri dish. These gene therapy vectors, when we inject them in the eye, you can see the red in the middle. They only go to the optic nerve head. So we've got a great tool tool for gene therapy for the optic nerve head for glaucoma. Now, what are we going to deliver? Well, here again, we made like a really neat discovery here. It turns out that there's a molecule inside cells called cyclic AMP. Anyway, cyclic AMP, it turns out, is the key molecule telling these astrocytes whether you should be good or bad in glaucoma or other optic nerve injuries. And so we can actually deliver gene therapy vectors that either increase the cyclic AMP or decrease the cyclic AMP. And when we do that, in these kinds of data shown here in animal experiments, we can either increase retinal ganglion cell survival after optic nerve injury or decrease retinal ganglion cell survival after optic nerve injury. And by delivering, again, these kinds of enzymes using this optic nerve head specific viral vector in an animal that has glaucoma, we can save not just the retinal ganglion cells that live right next to the optic nerve head. We can save all the retinal ganglion cells in the animal's entire retina. So we are super excited about this. We're calling this, I didn't make this word up, gliotherapeutics. So instead of neurotherapeutics, let's have gliotherapeutics. And by the way, maybe one day we'll put them both together and it'll be like a belt and suspenders for all of our patients. Okay, so how are we going to translate this out of the lab into the clinic? We need good ways to measure. We can't do clinical trials if we're going to sign everyone in the audience up and say, by the way, we'll follow you for 5 years with your visual field tests and see who gets worse. It's just too long, it's too expensive. We got to figure out better ways to measure glaucoma in our patients with glaucoma. A lot of people in this room are familiar with the visual field test. You put your chin on that. It's annoying. Yeah, exactly right. It's hard to do. You got to focus. You got to schlep into the office to do this. Good luck schlepping into the office more than twice a year. It's a pain, right? But to really use visual field, we got to be measuring your visual field. You know, like if you've got bad glaucoma, we want to do that every week. How are we going to do that? We can't have you come in every week. Any of you who have been to the Byers Eye Institute know between 9:30 and 3 o'clock, good luck getting a parking space. Never mind an appointment. But we've got companies now, startups, who have solved for that. For example, Peripherex uses eye tracking on your home computer, the same computer you might be Zooming your kids or grandkids with. It's using that same built-in camera, but to track your eyes and do a visual field test, it allows you to do visual field testing just in the home, over the computer. They did a FDA, registration trial. It showed it was, really remarkably similar to the in-office visual field that we're all doing. So really high, all those statistics looked really good for it. So that's moving into clinical use. They're just launching now for clinical use now in the US. So it's a very exciting way to think about moving that forward. What else can we do? Well, it turns out that within our retina, some retinal ganglion cells fire when the lights go on, but others fire when the lights go off. There's actually different types of retinal ganglion cells in our retinas. And it turns out that it was discovered in animal models that the retinal ganglion cells that fire when the lights go off, we call them conveniently off retinal ganglion cells. It turns out that those are the ones who die first in glaucoma. In animals. So we've been asking, well, could this be true in humans? Could this be a new way to measure the disease? And so here's Tony Norsha. He's a professor in the psychology department, and he does brain-evoked potentials called visually evoked potentials. He's wearing the hairnet that we use on patients to measure how your brain is reacting when we show you visual stimuli. So it's like a visual field, except you don't have to focus and push the button all the times, that kind of thing. So you just sit there and watch the computer screen. And using that and some tricks of what the computer shows, we can actually separate the on and the off retinal ganglion cells. And what we've discovered is, like in mice and monkeys, human patients with glaucoma also lose their off retinal ganglion cells first. And so we see this difference between off and on in the normals, and the glaucoma patients lose that off bias response very early in disease. We've got ways to structurally image the optic nerve. This is another picture of a healthier looking optic nerve. It's got more of that orange rim around it. We've got all our typical metrics that we like to look at when we're seeing you in the clinic. But now we can use high-definition imaging and look at every layer of the retina, so much so that we can start to look even at the metabolism. So we can now measure the mitochondrial health in your retina non-invasively with a simple picture. And we're using this in our clinical trials now at Stanford. And the idea with all of this is to measure disease before it's too late. Visual field catches you pretty late in the disease. By the time we detect something on visual field, you've already lost half of your retinal ganglion cells in that area of your retina. We need to look for things like loss of metabolic function or really the substructures inside the retina to measure it before it's too late. And the idea here is that instead of taking a long time to do a clinical trial, here in the red circle, we can look for things that change quickly, like your retinal healthy metabolism in response to a new therapy, and do clinical trials really quickly. And so that's what we're now leveraging at Stanford as we now are moving science over these last few years from the lab to the clinic. And this is the last piece I want to leave you with, is really the hope for this future that we're already bringing into now. This is not just hope for the future, this is now. And we at Stanford, and just starting to see also a few other places, but I want to give credit to our faculty and staff where this has really been a main focus for us, is starting to do clinical trials in vision protection and vision restoration at Stanford. So for example, one of those nerve growth factors called nerve growth factor, NGF, we tested that, delivered as an eye drop, and in the patients who got the real drops on the right, we protected, actually slightly improved, more of their optic nerve head anatomy. That's what the green is compared to the red. When we use those drops, even just for 8 weeks. There's another neurotrophic factor called CNTF, and that's in phase 2 now, and that's actually delivered as a little surgical implant, just slips right in the white part of your eye. It stays inside the eye for the rest of your life, making CNTF. And again, we have early data now from our phase 2 trials showing that it can improve the structure of the retina in the way that I just mentioned, but it also protects against degeneration. So it protects patients, even measured over just 1 year. We were able to show that it protects patients from losing bad spots on their visual field in a phase 2 trial. We're pushing right now to turn that into a big phase 3 trial in more patients, make that more widely available. Here's one such patient. In the left side of the screen is their eye with a very flat structure in the retina that didn't improve. But on the right, you can see how it went up. Their structure actually got thicker again, like more healthy in response to the CNTF implant. And what we see on the visual fields, normally in glaucoma, visual fields never get better. But in this patient, you can see the line trending upward where the visual field is actually improved in this patient who got the CNTF implant in that eye, but not in their other eye. Other things we've got to do, we've got to protect the cells within the retina. There's a drug called anti-complement C1q. We did the first trial in human patients, in glaucoma patients, to show that this anti-C1q can mop up this complement protein that we know is bad for glaucoma, that it's part of the cause of the death of the cells in glaucoma. There's another signaling pathway involved in the death of the cells called FAST and FAST ligand. Again, the first data are coming out right now on testing this, not just in animals, but moving into humans and showing some promising data in Australia so far. I mentioned those growth factors, and I mentioned that those growth factors work even better if you're stimulating. We first tested this with visual stimulation, where patients actually wear these virtual reality goggles, and we show them pictures inside the goggles that really overstimulate your retina. And we detected a little something, but not that much. So we actually reverted to testing electrical stimulation. So now we're doing clinical trials where we put little electric pads on your temples, not enough for like shock therapy, but just delivering a little bit of current across the retina. And again, we're seeing some really promising data that if we treat for 2 months, we can see some positive effects in patients out to 8 months, which is as long as we've tested so far. We tried boosting activity with hyperbaric oxygen, and so we had patients come in and actually lay down in one of these oxygen tanks, and they could read their iPad for an hour or listen to music, and we pumped extra oxygen in there. And at least in the most severe patients, it didn't have a positive effect. but we were also using like a slightly weaker version of the hyperbaric oxygen. So not sure yet. We want to return to that kind of work, still to come. And then the last couple I think are super exciting. One is vitamin B3, B as in boy 3, like nicotinamide it's called. And this has been really exciting because 2 phase 2 trials, one in New York and one in Melbourne, have shown that if you take high-dose vitamin B3, you can actually give a little boost to your metabolism. B3 works in part at those mitochondria. Give a little boost to the metabolism and actually improve the visual field in glaucoma patients. We're now doing, with New York, a phase 3 trial of high-dose vitamin B3 to see if we can really prove that. And I think very exciting, Young Hu, who's one of our faculty right here, has actually discovered a gene therapy that should work even better than taking the high-dose vitamin, or at least in concert with the vitamin. So it's a really exciting moment right now about sort of taking the experiments out of the laboratory and really trying to push them into human testing. And last, I'll just mention insulin eye drops, the same insulin that diabetics use for their diabetes. If instead of injecting it under your skin because you have diabetes, instead you drip it in your eye as an eye drop. We tested this for the first time last summer in glaucoma patients, and we found that the patients on the left who got the insulin, showed an improvement, a thickening of their nerve fiber layer. That's the peak in the middle. Then after they stop the insulin eye drop, it goes back to where they were before. So it showed that the insulin, whereas in the right side in the fellow eyes that didn't get an insulin eye drop, no change, no net change. So we're now in the middle of seeing, hey, can we turn this from like having to draw it up in a syringe? Can we put this in a bottle so you could just use it as an eye drop, make that easier? We're also testing like, could we give 2 drops a day and make it work better? Could we give 3 drops a day, make it work even better? And we're going to start a big clinical trial on that later this fall or early winter. And some of that work, I want to point out again, was funded through the donor network, through the Glaucoma Research Foundation. So this is how we are really pushing this forward. I like to also point out that by doing these trials, we get real world, real patient, real trial data, and every time we do a study, we learn something about how to do the study better the next time, and hopefully also inspire our colleagues around the country, around the world. Hey, you can do these studies. You start testing your drug of interest too. Let's all move this field forward. So we've got great therapeutic candidates coming out of the laboratories, and we're showing for the first time over these last few years that neuroprotection and neuroenhancement therapeutic candidates can be studied in our patients, and I really feel like we're moving from fantasy to reality. So I want to thank all of you, and I want to thank the Glaucoma Research Foundation for having me out tonight.